Hi, everyone. It's my pleasure to present in HBHL Research Day. This presentation is about increasing the genetic diagnosis yield in rare Mendelian neurological disorders. Hereditary spastic paraplegia, or HSP, is a good example of rare Mendelian neurological disorders, which is the umbrella term for a group of rare heterogeneous neurodegenerative monogenic disorders characterized by bilateral lower limb spasticity and weakness, with or without additional symptoms such as seizure, intellectual disability, and so on. Unfortunately, there is no treatment for HSP so far. The emergence of next generation sequencing revolutionized the genetic diagnosis of HSP through the discovery of over 80 genes or loci. Each gene or locus causes a distinct HSP subtype. CanHSP is a collaborative effort and consortium of 7 major centers across Canada to create a national database for HSP. It consists of more than 700 HSP cases from about 400 families and is one of the world's largest cohorts of HSP. Despite the use of whole exome sequencing, a genetic diagnosis cannot be made in more than half of the HSP cases. In our cohort, the results are consistent, and we couldn't identify any potential disease-causing mutations in 42% of families. So there are potential explanations to account for the large fraction of unsolved cases. Firstly, whole exome sequencing cannot identify certain changes, including deep intronic variants, structural variants, such as CNVs, repeat expansions, which all were reported as disease-causing in similar disorders. In 2019, deep intronic POLA3A gene variants confirmed as a frequent cause of HSP. Secondly, some variants are in genes not yet linked to any disease, as we call new genes. Our team members identified calpain-1 mutations as a major cause of HSP in 2016. Since then, more than 10 studies have reported HSP patients with calpain-1 mutations. In 2019 alone, we and other teams identified biallelic variants in a potential new genes as a cause of for HSP. The third explanation is because HSP is usually considered to be caused by monogenic Mendelian mechanisms. However, this oversimplified view is not necessarily true. For instance, oligogenic inheritance, repeat expansion, reduced penetrance alleles, modifier alleles were all reported in HSP and similar disorders. More recently, it was demonstrated that the EXOC4 gene may be involved in complex inheritance in axonopathies, including HSP and CMT. So the main objective of this project is to fill the diagnostic gap and better understand HSP. Specifically, the current research project focuses on deciphering the genetic causes of HSP using NGS approaches. We hypothesize that studying a large and Canada-based cohort could help to identify the currently known and unknown genetic variations in HSP. With the identification of new genes, of course, researchers around the world can add them to their panels of HSP-related genes for sequencing, possibly resulting in correct diagnosis. It may also lead to designing new drugs. Moreover, reaching the accurate diagnosis would be beneficial for HSP families and could prevent the disease in additional generations, leading to a reduction in the socioeconomic burden of HSP. Finally, This research was undertaken thanks in part to funding from the Canada First Research Excellence Fund, awarded to McGill University for the Healthy Brains, Healthy Lives initiative. This project was also financially supported by FRQS and MNI awards. Thank you.